Okay, welcome everyone. Now we are in the week 8, which is on nucleic acid. So I hope you all have gone through the lectures that ma'am has uploaded on NPTEL portal. And week 8 talks about nucleic acid, and there were total 3 lectures on nucleic acid 1, nucleic acid 2, and nucleic acid 3. Those who are joining on YouTube and who will be watching the recording later on, for them, let me introduce this course. This is NOC 2023 CY30 Biochemistry course, and I am Karan Kumar, Senior PMRF Scholar at IIT Guwahati, and I take regular weekly live sessions on this in this course. So about me, this is my email ID, karan18@iitg.ac.in. You can ask your doubts or any relevant question. If you have, you can email that to me. And my website is www.iitg.ac.in/stud karan18. And you can watch my all recordings of the previous lectures on YouTube. My YouTube link is www.youtube.com/winYOCV2022. So you can redirect to this YouTube channel, and then you can watch all the related recordings of the previous sessions. Now, coming to the week 8, the concepts that were covered in week 8 were definition of nucleic acids, and then ma'am talked about the structural units of purines and pyrimidines. Then conformation of sugar rings, in which she talked about N and S twists. Then torsion of the bond rotation and nucleotides. Then she talked about the formation of nucleic acid chain. Then types of base pairing, how many types of base pairings are there, and then tautomerism in nucleic acid bases, senses and anti-senses strands of DNA, and double helical structure of DNA. Then she talked about 3 different type of DNA, then types of RNA and its elementary structure. As you all know that there are 3 DNA which are A, B and Z type. Then she talked about how to make the table for standard genetic code, then factors promoting the DNA stability, hydrolysis of DNA, which is crucial to understand when you want to replicate your DNA, and if you want to modulate the renaturation and denaturation of the DNA. Then endonuclease, then exonuclease, and along with their function. Then DNA renaturation and denaturation, both ma'am talked about, and then the factors that affect the DNA denaturation and renaturation also ma'am talked about. Then the role of melting temperature, the definition of melting temperature and the associated definitions. A few general questions I would like to ask on this current topic is, first question is, what is the role of nucleic acid and why is it so much important in biology? Why do we want to understand nucleic acid, and what is the scope of this with research perspective? So, can anybody unmute themselves and then answer this question? What is the role of nucleic acid? Anyone here who want to answer? Okay, I will wait for few minutes and then I will answer this question. What is the role of uh, nucleic acid? Till then, let me correct the slide. You can unmute yourself and answer the question. Who all are here? Okay. Pragna, do you want to try this question? What is the role of nucleic acid? Why is it so much important in biology? I hope you have come like read gone through the lectures that ma'am has uploaded on NPTEL portal. So anyone? So there are two types of nucleic acid, RNA and DNA, and both have their specific role. Within RNA, if you go, you will have rRNA, you will have tRNA, and you will have mRNA, right? And there are different role of this RNA in the biological system. rRNA makes the ribosome and they help in the protein folding, protein formation, and other work. tRNA stands for transport RNA, and transport RNA helps in translocating the codons or transferring the RNA molecule from one place to another place so that it can be translated to amino acid. It can be translated from amino acid system to the protein, and then mRNA is the messenger RNA. Okay. Coming to the role of DNA, DNA plays a very crucial role. This is the genetic material of the any biological system. Genetic material. What do you understand by the term genetic material? What is genetic material? Anyone? Okay. Genetic material means while when the cell is dividing or when cell is going to form its progeny, then during that time it will replicate, it will try to make its own progeny by its own DNA or the genetic material which will be transferred from one level to another level, means from mother or parental DNA to progeny DNA. And how the cells do that? They replicate their own DNA, and then during the cell separation or the cell division, this DNA is being this DNA is being equally divided into the two cells. That is what is being done in the bacterial cell. And in human cells also, during the mitosis, the cells are being replicated in this fashion. Okay, the chromosomal DNA gets divided into like equally, it divides into two cells. Why is it so much important in biology? Because whatever like the whole organism can grow from the DNA itself, because all the informations that a organism will develop or grow, that all the information are stored in DNA. Now the question arises, when the nucleic acids are of two types, RNA and DNA, then why DNA is most favorable and RNA is not? Why cells wants to store its information, or why nature has designed DNA as a storage material, not RNA? Do anyone want to answer this question? Why DNA act as a storage material, storage information, not RNA? Initially, there was a term like when there was debate going on, like whether RNA is genetic material or DNA is genetic material. Initially, it was thought that RNA is a genetic material. How they found it? Like, and that time it was like called as a RNA world hypothesis. So in RNA world hypothesis, how they act as that they try to answer that RNA in the RNA world hypothesis, they try to answer that basic problem that what is self-replicating, because RNA can be self-replicating because all the informations that it needs to replicate are stored in RNA, and that is true in some of the viruses. Like in viruses, when the virus is being replicated in some of the viruses, the RNA is the genetic material. They don't have GNA in their cells, and they kind of double, or like propagate with replicating their RNA. So initially, when the TMV virus, tobacco mosaic virus, was discovered, then what they saw that RNA is the main material which is available there, not DNA. Okay, and it was self-replicating. So then at the scientists thought at the beginning of life is nothing but RNA. Okay, but this hypothesis that was proposed during 19 in sometime in 1986 or before that, in the like when the Gilbert gave this hypothesis of the phase RNA world. So this time they supported the RNA as an ability to store. But why RNA cannot store is that at the 2 prime, when you see the like sugar phosphate of the RNA, which is ribonuclease, the difference between RNA and DNA is that in DNA you have deoxyribose. The both full form is the RNA is ribonucleic acid, and DNA is deoxyribose nucleic acid. That means at some of the position, the oxygen will be not present in DNA. Like if you see, and the numbering is being done through this 1 prime, 2 prime, 3 prime, 4 prime, and then 5 prime. Okay, at the 1 prime end, you will have a nitrogenous base, and at the 5 prime, you will have phosphate bond, phosphodiester bond. To phosphate group, and at the 3 prime your like it will extend to the another chain. But the difference between the ribonucleic acid and deoxyribonucleic acid, in the ribonucleic acid you will have oxygen hydroxyl group at the 2 prime. However, in the deoxyribonucleic acid means DNA, this RNA will have 2 prime. At the 2 prime, they will have hydroxyl group. However, in DNA, it is not present. In DNA, it is present as only hydrogen. That means the oxygen is not present. This is the extension. Now here you will have the phosphate, sugar phosphate bond. This is the nitrogenous group. Okay. Now the difference between the DNA and RNA is this. Now how it helps? Because of this, because of the presence of OH, it if you alter the pH, then this OH can act as a nucleophilic attacker, and that's how it can denature the like whole chain. So that is why RNA is not stable in the nature. However, DNA lacks this OH group and it has only hydrogen. Because of this, it can form a double stranded form. It can be in the double stranded form, and also it is more stable than the RNA. Okay, because of this, difference with at the 2' end. This is also 2'. Okay, so that is why DNA is more stable genetic material. Okay, now what is the scope of this topic with research perspective? There are lot of research going on in this area, and it is from the early days till now. People are still surprised with the complexity of DNA. DNA structures have been solved by Watson and Crick model they have proposed, and but still this uh, like uh, why, how, and these all things have not been solved yet. Thereby, uh, like uh, doing the mutations and the other things, the research area is still going on in this area. You isolate the DNA based on the information that you have got from the computational biology, that if you mutate this particular site, or the if you mutate this particular gene, then the gene replicate, while gene will transcribe into the mRNA and then to the protein. When it translates to protein, then if you make the changes at the genetic level, it then that will be translated to the protein level. So this is how the research is going on in this area, and this is a very fascinating area for research. Now, what type of question are asked in KIT question? So mostly numerical type of questions are asked from this topic, in which some of the problems I will discuss in the later slides. Now, let's go through the assignment problem. The first question is, which of the following statement is not true regarding the double helix of the feature and the features of Watson and Crick pairing? Your statements are: the combination of staircase sugar phosphate backbone and the bonding between the base pair results in a double helix. Statement 2: 5' to 3' strands is known as the sense strand, whereas 3' to 5' strand is known as the antisense strand. Statement C: the dimension of the two permitted base pairs in the Watson and Crick model are similar. Statement 4: the original Watson and Crick model was based on the aDNA, which is the predominant form of the physiological condition and it has 10.5 bases per ton. So, anyone want to answer this question? Which of the following statement is not true?
Option D.
Option D. Okay, then what is the correct answer?
bDNA is the most predominant and found on physiological condition.
Very good. So option D is not correct. However, other three, the 5' to 3' strand is known as the sense strand. That is true. And 3' to 5' is known as the anti-sense strand. What is the lagging strand and leading strand? Which one is the leading and lagging while replicating the genetic material?
The strand that is continuously formed is leading strand.
Okay,
Whereas this,
and
which is
which direction it is replicating continuously? 3'
3
to
prime,
5' or 5' to 3'?
3 prime to 5 prime.
Very good. So here the correct answer is D, which is not correct. And the combination of the staircase sugar phosphate bond and the bonding between the base pair results in the double helix. This statement is also true. And what is the makeup of DNA? Like DNA or RNA, nucleic acid. If you if you disintegrate the nucleic acid, which all things will you get? What is the composition of nucleic acid?
Base and sugars.
Which base, which sugar? What is the generic name?
Nitrogenous base and in DNA there will be deoxyribose sugar. In RNA there will be ribose sugar.
Okay, so basically it is sugar. Basically it is sugar, which is pentose sugar, right? Normally you find only ribose, right? Ribose. In case of DNA, it will be deoxyribose. In case of RNA, you will have hydroxyl group at the 2 prime. So one thing you will find sugar, another thing you will find phosphate group, and then what you will find nitrogenous base. So what all are there nitrogenous base? Which all nitrogenous base you know? What is the 2 category of nitrogenous base?
Purines and pyrimidines.
Purines and pyrimidines. And which all are purines and which all are pyrimidine?
Adenine, guanine are purines, and whereas cytosine, thymine, and uracil are pyrimidines.
Very good. Very good. So there are total 5 bases termed as the nitrogenous bases, and these are categorized as purines and pyrimidines. Normally in purines, you will have 2 fused ring, right? One is six carbon ring, and another is 5 carbon ring. These will be the fused. And in pyrimidine, there are totals 6 edges, like 6-member ring. Now the next question is 3 different segments of A DNA, B DNA, and Z DNA contains a total of 1320 base pairs. What is the approximate length of the longest DNA possible? Based on the above statement, can you identify 3 different segments of ADNA, BDNA, and ZDNA all contain 1320 base pairs? So what is the approximately length of the longest DNA possible? Can somebody calculate it and tell me how you done it?
495.
495. How you have done it?
That DNA is the longest form of DNA, and it contains 12 base pairs.
12
So,
base pairs in one turn, right?
yes, sir.
Okay.
So, if total base pairs are 1320, we will divide by 12 to get how many
Okay.
turns are total. Yes,
But
sir.
that will give you the only in turn, right? What will be the length?
Then,
How
then,
you
we will,
calculate the length?
then we will multiply with the per turn length.
So what is the per turn length?
It is 3.9 nanometer.
Okay. So for ZDNA, because out of A, B, and ZDNA is the longest form. Like it is the most relaxed form, right? So it is how many per turn, how many length, what will be the diameter of it? Stretch, length in stretch. What will the stretch? 3 point? What will be the length of per turn for ZDNA? Yes. So there are total 1320 base pairs. Okay. And in ZDNA, you will have 12 base pairs, 12 base pairs in 12 bases in one turn, right? So you divide it with that and then multiply with the number, the length in one turn. Okay. And that will give you the length of DNA in one turn, and that will give you the length of longest DNA possible. Okay. Anyone faced difficulty in solving this problem? Anyone who have joined here and they faced problem in solving this problem? Like, did they face any problem while solving this question? Or is it clear now? And this is the basis of solving all kind of question. Like if you, even if you don't know, like the basic formula you need to remember. If you have given any kind of base pairs, then you first put, like if you want to calculate the length of DNA in nanometer, then what you have to do, how many base pairs you have given. So number of base pairs, okay, number of base pairs, then you divide this by the how many like how many turns are there in one base pair. Okay, okay, in one turn base pairs in one turn, then you multiply this with length of one turn. So if you fill in these details, and this length of one turn will be in nanometer, right? These two are in base pairs you need. So these base pairs will cancel out, and finally you will have the length of DNA in nanometer. Now you must remember, like you must cross check your what is your kind of unit you have been asked. So in the question it was asked in nanometer, so you put the length of one turn in nanometer. Normally it is given in the angstrom. So if you have been given in the angstrom, then you convert that into the nanometer. Angstrom means 1 to the power 1 into 10, 1 to 10 to the power minus 10. That is the meter. That is the angstrom. In nanometer, that will be 10. 1 angstrom is equal to 0.1 nanometer. You must take care of this unit conversion so that you don't fall in trap and then you miscalculate your answers. Okay, so what was the answer for this question? What was the answer given in the question?
Option A, sir.
Option A. Very good.
So, correct
Yes.
length 4.5 nanometer. I said 3.2. That is not for ZD.
Yeah, that is why I was asking again and again. And I did not answer this one because I wanted you all to recheck it. The
Nice.
correct length of is 4.5 nanometer in ZDNA. Okay.
Nice.
Now the next question is, what should be complementarity strength of this given strand? Your given strand is TACGG AACT, and this is 3 prime. This is 5 prime. What will be the complementary strand?
Option D 3 prime AT GCC TT GA 5 prime.
Okay, option D. Now, how many hydrogen bonding is being formed between A and T?
2, sir.
Yes. In A and T, you will find 2 hydrogen bonds. However, in G and C, you will find 3 hydrogen bonds. Why is it? Why is it so? Why is it so? Why A forms only 2 hydrogen bonds and G forms 3 hydrogen bonds? Any explanation of it? Which all groups can participate in which all atoms can participate participate in hydrogen bonding? And why?
Nitrogen.
Yes.
Nitrogen and oxygen.
Nitrogen and oxygen. And why only these two can participate in hydrogen bonding? Why there is a hydrogen bonding in first place? Tell me that. Why two atoms makes hydrogen bonding? Yes, you were saying something?
Because of their electronegativity.
Yes, because of their electronegativity. The normally when we make the hydrogen bonding, then what we do that we put hydrogen in between that, but that is not true. Normally, this hydrogen will be more attracted to the more electronegative atom. In this case, the oxygen is the more electronegative atom. So hydrogen will be partially attracted towards the oxygen. Okay. So what do you and why the hydrogen bonding is being formed? That is because of the electronegativity. So yeah, and this hydrogen bonding is nothing but the generation of partial charges on the atoms, and in that fashion the electrons are being much more polarized towards the which is the most high electronegative atom. So in that case, now if you see the structure of guanine, cytosine, adenine and thymine, let me go through that. Adenine and thymine. See, if you see in adenine and thymine, the two groups that can form the hydrogen bonding is the first is this nitrogen of the adenine, this nitrogen and thymine. So the two nitrogen bonds, the two nitrogen can also share the hydrogen. Now one can ask that this particular both the nitrogen have the same electronegativity negativity, then why they can share the uh, why how they form this hydrogen bonding? This hydrogen bonding is being formed because in this case, due to the delocalization, the lone pair does not participate in this delocalization. However, the hydrogen attached to this nitrogen have lesser will have the higher electronegativity than this nitrogen. Okay, so because of that, there arise a electronegativity difference. Because of the lone pair is not denucleizing in this ring and it is not fused in this ring. So in that case, this nitrogen will have less electronegativity compared to this, and thereby the hydrogen will be shared between these two. However, in thymine, this oxygen will contribute towards the more electronegativity to this hydrogen, and there how they form the two hydrogen bonds because they have only two sides. But that is not true in case of this GNC, which is guanine and cytosine. In guanine and cytosine, you will have three hydrogen bond formation because of their structure that exists in their like the atoms that exist in their structure. Okay, so that is what is being represented here. So A and T forms two hydrogen bonds, G and C forms three hydrogen bonds. Okay, now let's come to the next question. The question is,
excuse me, sir.
yes.
Is this meeting about week 7 or week 8?
This is week 8.
Oh, sir, I have a question of week 7.
Yes. Which is your
13th
question?
question in the assignment, the
Let me
thermodynamics of lipids.
Thermodynamics of lipids. Okay. Fourteenth questions.
13th.
13th. Okay, yeah.
How to solve it?
How to solve it? Okay. So this I discussed in the last interactive session. Yeah. So you can solve this question by the formula that was given as a chemical potential. Okay. So in chemical potential formula, delta mu is equal to RT ln. Okay. RT ln. The formula was given. Let me give you that formula. I had uploaded the slide also on the drive folder. See, this is the formula for solving this kind of question. So chemical potential delta mu is equal to RT ln C2 by C1 plus Z F and V2 minus V1. So in this question, what all information you have been given? In this question, you have been given. Okay. You have been given V1 and V2, like calcium ion. The C1, C2 is given. Concentration in one compartment and concentration in second compartment is given, and you need to calculate V2 minus V1, right? If you fill this information in this formula, like Z you have, Z is nothing but the charge it can have. On calcium it is +2, right? Calcium is +2 ionized, so Z here is equal to 2. Now Faraday, this is the F is the Faraday constant, which is 96500 calorie per mole. Okay. Now V2 minus V1 you have to calculate. C2 you know, C1 you know. Then R is gas constant. Temperature you have been given 25 degrees Celsius. Delta mu you also know, which is 9000 joule per mole. Now the only thing that you have to consider that about the units. If you carefully examine the units, which is R is given in the joule per Kelvin per mole, and the temperature here is the 25 degrees Celsius. So that means you have to convert the 25 degrees Celsius to Kelvin. Then in the R itself, it is like all the both of them have been given in the joule, but your Faraday is given in the this calorie. So you have to convert this calorie to the joule and then fit these all information in this formula. Then you will be able to get the answer. Your correct answer will be the 12.5 millivolt V2 minus V1. Will you be able to solve now?
I will solve it and tell you. Thank you.
Yeah, yeah. So if even if you get any difficulty in solving, you can ask in the meeting. Okay.
Yes.
So the next question. Anyone else has the question from week 7 or the previous assignments? Anyone else? So the formula was delta mu, which is chemical potential, RT ln C2 by C1 plus ZF V2 minus V1. If you apply this formula, you will be able to solve the question number 13. Okay. The next question is the rate of hydrolysis is slower in DNA than RNA. This is because which of the condition? Option A, the ribose sugar present in DNA is in deoxy form, but it is not so in RNA. Option B, of the presence of different pyrimidine in RNA compared to DNA. Option C, the stacking of nitrogenous base. Option D, the phosphate in the backbone of RNA favors hydrolysis at faster rate. Which is the correct option here? I had in the beginning itself, I had disclosed this. I had discussed this. Why rate of hydrolysis is lower in DNA than RNA? Anyone? Who all are here? Anyone want to answer this? Okay, so that I had discussed in the beginning of the lecture that DNA, which is in term, if you elaborate this term itself, then you will find the answer to this question, which is DNA stands for deoxyribose nucleic acid. However, RNA is ribonucleic acid. So now in DNA, the one of site is deoxy form, which is 2 prime. In 2 prime position, instead of hydroxyl group, which is present in RNA, you have hydrogen in DNA, which is in the deoxy form. Because of that, the hydrolysis is slower in case of DNA. However, when you come to the hydrolysis, this OH will act as a nucleophilic attacker on itself, the RNA structure, and that is why the hydrolysis occurs more faster in RNA than DNA. Because in DNA, you will get a deoxy form at the 2 prime. However, at the 2 prime, you will have hydroxyl group in RNA. Okay, so option A was correct. Now the next question, dash in ribonucleotides renders RNA molecule susceptible to the strand cleavage in solution. This is also the same thing that we discussed. In which case the oxygen hydroxyl group will be deprotonated. Alkali or acid condition. Anyone? Why alkali or why acid? The answer I already given about the position, that was 2 prime. Now you have to choose why alkali or acid. In which solution the RNA molecule will be susceptible to the strand cleavage. Anyone?
In alkali medium.
Am I audible? Anyone?
Sir, in alkali medium.
Alkali, why?
Because it has OH for the ribonucleotides to be dismantled.
No, but in acid condition also OH will be there. So why alkali? Why alkali is why in alkali solution RNA molecule will be more susceptible. The 2 prime OH will be in both of cases, right?
So in the medium.
In the medium, yes. Alkali will act as a base, and because of this base, because it has in alkali, you will have, in case of hydrogen H plus ion, you will have more like OH minus ions, right? More basic medium. So this alkali will subtract will abstract this hydrogen from the OH group, and then this O minus will act as a your nucleophilic attacker, and because of that, the RNA molecule will be more susceptible in case of alkali solution. Option A is correct here. Now the next question is possible favored conformation of phosphate in phosphodiester bond occurs at which angle? Option statements are Gauss angle, which is plus and minus 60 degree, then extended 180 degree, eclipse 0 degree, and anti 180 degree. Options are the possible favored conformation will be 1, 3, and 4. Option B, third and fourth. Option C, first and second. Option D, first and third. This is factual question, so you need to remember this. Normally, yes. Anyone? Normally, the favored conformation in the phosphate of the phosphate in phosphodiester bond is the favored conformation is Gauss and extended. Okay, so option C is correct here. Okay. Now the next question is yes.
So,
yes.
What is Ln in the thermodynamics formula?
L?
L and N in that lipid thermodynamics formula.
Ln and I didn't got anywhere ln. The formula that I have given you, delta mu. This one?
Yes, sir.
In this, there was no L and M. This is mu. Mu is equal to RT ln. This is logarithmic base E. This is not ln. Like you write log, right? Log is the base 10, but ln is the log of like in which base is E. When in case of log, when base is E, then it is termed as the ln. This is not L and N separately. Okay. This is natural logarithm of C1 by C2. Okay. So the question like here in this question, the correct answer was C. Now the next question is which of the following option does not correspond to the pyrimidine base found in RNA? Statements are uracil and thymine, thymine and cytosine, adenine and thymine, and cytosine and uracil. Which does not found in which of the pyrimidine base does not found in RNA? What do you find in RNA? What is the composition of DNA? And what is the composition of RNA in terms of nitrogenous bases? Somebody want to answer? In DNA, you have A, T, G, and C. However, in case of RNA, you have A, U, G, C, in which in place of T, thymine, you have uracil. The question was which is not correspond to the pyrimidine base found in RNA. Now, out of this, which is the pyrimidine and which is the purine? Anyone? What are purine and pyrimidine? Purine is adenine and guanine. This question was discussed earlier also. Purine is adenine and guanine, and pyrimidine is cytosine, thymine, and uracil. Okay. Now, in case of RNA, which one is not found? This adenine and guanine, these two will be your purine. You are talking about pyrimidine. Okay, so uracil and thymine is obviously there. Uracil and thymine both are like uracil is pyrimidine, but which is not correspond to the pyrimidine bases found in. Your correct answer will be option D. Okay. The next question is identify the correct statement regarding the nucleic acid structure. Regarding the nucleic acid structure, statements are nucleic acid are made up of polymer of 4 different nucleotide residue each. Statement 2, among these, RNAs uses AMP, TMP, GMP, and CMP. And option 3, DNA uses deoxy form of DAMP, DCMP, DGMP, and DTMP. Which of the statements are correct in case of nucleic acid structure? Options are 1 and 2 is correct. Option B, 2 and 3 is correct. Option C, 1, 2, and 3 is correct. Option D, 1 and 3 are correct. Any answer? Obviously, statement 1 is correct, right? Which is talking about the nucleic acids are made up of polymer of 4 different nucleotides, which is correct in case of both DNA and RNA. For DNA also there are 4 nucleotides, and for RNA also there are 4 nucleotides. In case of thymine, you will have uracil. Okay, and now talking about the amongst RNAs use AMP, TMP, GMP, CMP, which is incorrect. Statement 2 is incorrect. Now option 3, like statement 3, which is DNA uses deoxyform, DAMP, DCMP, DGMP, DTMP. Yes, this statement is correct. So option D is correct here. First and third. Now coming to the next question. Which among the following statement is or are false about the DNA melting temperature? Statements are the higher the salt, higher the TM. Statement 2, the higher the GC content, lower the TM. Statement 3, low, which is less than 2.3, and high, which is greater than 11.5 pH, decreases TM. Which statements are false regarding DNA melting temperature? DNA melting temperature is nothing but TM. TM is nothing but at which the 50% of DNA is denaturated. TM is the melting temperature at which PM is the melting temperature at which 50% of DNA strands denaturated, okay, or unzipped. So which all factors kind of determine the DNA melting temperature? Of course, all three content will do, but in which fashion? Higher the salt content, higher the TM. Higher the GC content, lower the TM. And option like statement 3, low pH and high pH decreases TM. Anyone? Your correct answer here is 2 only. How? If you increase the salt content, you are here to find the false statement. So if you increase the salt content, that will stabilizes the like hydrogen bonding and the like salt content such as Mg2 plus. If you increase the Mg2 plus, it will increase the bonding and it will make the nitrogen species more stable. So higher the salt, higher the TM. This statement is correct. Now low pH and high pH both decreases the TM. This is also correct because in both of the condition, your hydrogen bonding pattern will change. If you decrease the pH, then also your hydrogen bonding number of hydrogen bonds will decrease. And if you increase the pH, that time also your number of hydrogen bonding will decrease. So if you decrease the number of hydrogen bonding, that means your melting temperature will go down. That means your DNA will be more unstable. So statement 3 is also correct. But in case of higher GC content, because adenine and thymine makes 2 hydrogen bond and guanine GC makes 3 hydrogen bond. That means if you are increasing the GC content in some stretch of DNA, if a given DNA strand, you have been asked that this particular strand has higher GC percentage, GC content. That means it will have high number of hydrogen bonding. If you are increasing the number of hydrogen bonding in a particular DNA stress, it will take more time to denaturate, right? Like more number of hydrogen bonds to denature more number of hydrogen bonds, you will need more energy. You will need to increase more temperature, and that is why this statement is false. If you increase the GC content, your TM will also increase. It will not decrease. So only statement 2 is incorrect. Okay, option B is correct. Option B is the answer here for this question. Now coming to the next question, which among the following is or are false statement about the stability of DNA? It is the same as the previous question regarding the stability. Previously it was denaturation. Right now it is about the stability. Your statement 1 is charged and hydrophilic sugar phosphate backbone are in the interior. Option 2. Statement 2. Stabilization through base pair hydrogen bonding is greater than the stacking interaction. Statement 3. The hydrogen bonding guarantees the complementarity of the strands. Statement 4. Stacking interactions are sufficient to overcome the electrostatic repulsion of the phosphate. Which among the following are incorrect? Options are first, second, and fourth. Option B, third and fourth. Option C, third only. Option D, first and third. Which all are incorrect? And why? Anyone? Here, when we talk about the statement one, which is charged and hydrophilic sugar phosphate backbone are in the interior. Let us assume that if you watch the double helix, if you watch the double helix in like up from up, it will show you as a circle. What will you see? Because there will be hydrogen bonding between, like, this double strand is just because of the hydrogen bonding that is there exist in the anti-parallel strands of DNA, right? So because of this hydrogen bonding in the interior, you will find this hydrogen bonding patterns. But if the sugar phosphate backbone comes into the interior, then what it will result, this charge and hydrophilic backbone will disrupt, will have the steric hindrance with the hydrogen bond formation, and the DNA will not be stable. So your statement one is obviously incorrect. This charge and hydrophilic sugar phosphate backbones are protruded outwards, not in the interior. So this is false. Now consider statement 2. Stabilization through the base pair hydrogen bonding is greater than the stacking interaction. Is it true? Is it true? This is also false. This statement is also false. Why? Because if stabilization through base pair hydrogen bonding was greater than the stacking interaction, then the double helix formation would not be much stable in that case. Because stacking interaction is what that results in making the proper turns and this kind of like making the stack of the DNA. So normally stacking interactions are greater than the hydrogen bonding base pair, and that stabilization is much more in case of stacking interaction. So this statement is also false. Now talking about the statement 3, the hydrogen bonding guarantees the complementarity of the strand. That is very true, because until unless you have the proper kind of hydrogen bonding, then you won't get the double stranded form, and this hydrogen bonding can only occur in the complementarity. If it is the hydrogen bonding is occurring within the strand, then you will not get the double stranded form, and that DNA will be prone to denature. Okay, so you will not see that there is the intramolecular hydrogen bond formation. However, the hydrogen bonding is normally made into the complementary strand. So this statement is true. And stacking interaction are sufficient to overcome the electrostatic repulsion of the phosphate. That is false, because you also need hydrogen bonding. So which statements are false? First, second, and fourth. So option A. Now the next question is, the overall rate of DNA renaturation is determined by which all the statements? Option A, statement A, the concentration of DNA, the average length of DNA segment, and the complexity of DNA. Options are statement 1 and 2 are correct. Option B, only 3 is correct. Option C, 1, 2, and 3 are correct. Option D, only 1 is correct. Anyone? Anyone would like to attempt this question? How overall rate of the DNA renaturation is determined? If you recall that renaturation means if you remove all the denaturating factor, then whether the DNA will reform itself and renaturate, that condition is called the DNA renaturation. So obviously, what is the concentration of DNA depends. What is the concentration of DNA present in the solution? That will obviously affect the DNA renaturation process. So statement 1 is correct. It will be determined, like it will be involved in the DNA renaturation process. So correct statement. Now statement 2, the average length of DNA segment, that is also correct, because the more the length, it will take more time to regenerate, and more the length, it will have the more prone to make mistakes. Option 3, the complexity of DNA. Yes, that is also true. So your correct answer here was option C. It depends on all three factors. RNA polymerase is involved in transcription. Is it true or false?
True, sir.
True. Very good. RNA polymerase does involve in the transcription process. And what is involved in the replication? Which enzymes make the DNA replication? Obviously, it will be DNA polymerase, right? And there are different form of DNA polymerases. Okay. Now the next question is ATPs. Now the next question is ATP synthesis by ATP synthase is driven by which movement? Which of these three? Option A, H plus movement. Option B, NADH movement. Option statement 3, electron movement. Your options are 3 only, 2 only, 1, 2, and 3, and option D, only 1.
3 only.
Is in 3 only? Are you sure? ATP synthesis by ATP synthase is driven by which movement? If you remember the ATP synthase mechanism, normally you pump hydrogen outside of the of the membrane, and then based on that 4 hydrogen is being pumped outside, and then based on that it take the turn. The synthase machinery will take turn, and correct answer here is the H plus movement. Option D. Okay. Electron movement, electron is being captured by the oxygen and then it get reduced to the like water. Okay, that is the role of the oxygen. Oxygen will accept the electron, but that will be in the initial stage at the complex 3 only. But what occurs at the ATP synthase? In ATP synthase, during the machinery rotation, you pump 4 hydrogen plus ions, and then based on that, that energy is being converted to from ADP to ATP, and ADP is converted to ATP, during that process, and 4 hydrogen are being pumped outside the cell, outside the matrix. Okay, and that is how you synthesize ATP. So option 1 is only correct here. Now the question is, which of the following are false about the DNA topoisomerase? What is the role of DNA topoisomerase? Anyone remember? What is the role of DNA topoisomerase? Okay, DNA topoisomerase helps in releasing the revealing the strain that is caused by the replication. Okay, so it cuts the DNA, but that is, you need to know which isomerase cuts where. Okay, so your statements are: it releases the strain because of unbinding of DNA by helicase, relax the coil. Of statement 2, topoisomerase type 1 cuts both strand. Topoisomerase type 2 cuts one strand. Your options are only which of these are false. Options are 2 only. Option B 2 and 3. Option C 1 only. Option D 1, 2 and 3. All are incorrect.
So 2 and 3 only.
Very good. 2 and 3 is false. These two are false because the vice versa is true. Okay. Now the next question and last question, which is which of the following, which are the correct statement regarding the exo and endonucleases? After this question, I will help to solve you some of the numerical problem which are being asked in the previous year paper, and if you solve those problems, that will help you to gain the confidence while you encountered through some of the problem. Your last assignment problem is which are the correct statement regarding exo and endonucleases. Statements are: exonucleases catalyze the normal of catalyze the removal of terminal nucleotides 3' and 5' types. Statement 2, endonucleases catalyze removal of external phosphodiester bonds. Statement 3, type A, endonuclease act on the 3' hydroxide group of the nucleotide with the phosphorus group. Option 4. Statement 4, type B exonucleases act on the 5' hydroxyl group of the nucleotide with the phosphorus group. Statements are 1, 2, and 1, 2, 3, 4. All statements are correct. 1 and 3. Okay. So your statements are 1 and 3 are correct. Very good. So can you explain why? Can you explain why? This statement is obviously incorrect. It does not catalyze the removal of external phosphodiester bond. However, it cuts at the where does endonucleases cut? Where does endonucleases cut? Why do you think 1 and 3 are only correct? Okay, this is your homework. You revise this. What are the cleavage site of endonucleases and exonucleases? Let me move forward and discuss some of the important numerical problems that you might encounter in your competitive examination or in some of the like your end-term examination. So the first question is that I want to discuss. The first question that I want to discuss is I will take some exemplary problem so that you will be able to solve the similar problems. Your question is, what is the approximate molecular weight of duplex DNA required to code for glyceraldehyde phosphate dehydrogenase glyceraldehyde phosphate dehydrogenase whose molecular weight is 40,000. The enzyme which is glyceraldehyde phosphate dehydrogenase, molecular weight of this enzyme is molecular weight of this enzyme is, where is the cursor? 40,000. So how will you calculate this? How will you approach this problem? How will you approach this problem? How will you approach this problem? You have given a enzyme whose name is glyceraldehyde phosphate dehydrogenase, whose molecular weight is 40,000 Dalton. So what will be the approximate molecular weight of this duplex DNA that will be required to code for this particular enzyme? I will take another color to solve this question. Now you have been given a molecular weight. Where is the cursor here? Molecular weight of enzyme is nothing but this protein, right? So molecular weight of this protein is 40,000 Dalton, right? Now the question is, like, how do you get this protein? The first thing that how do you get the protein? There will be a gene, then that will be converted to the mRNA. Now this mRNA will be translated to the protein. Now the question is being asked at the protein level. So what you have to get? First you have to get how many amino acids will be there in this particular, what will be the average amino acid will be there in this particular protein. So how you will calculate that? For that, you need to know what is the average weight of amino acid, a particular amino acid. So you always remember this thing, average weight of amino acid is 110 Dalton. Okay, average weight of amino acid is 110 Dalton. So how many amino acid will be there in this particular protein? Number of amino acid? Number of amino
3.
acids? Not 3. That will be 40,000 divided by 110. So how many? That will be 300, approximately 364. Right? So how did I approach this problem? First, I will see. First, I will see how many how many amino acids are there in this particular protein. Your molecular weight is given. So average molecular weight of a amino acid is 110 Dalton. So number of amino acid will be 40,000 divided by 110, which is 364 amino acids. Now how you will go? You will go now, like in one amino acid, how many nucleotides are there? How many in codon, if you go, three letter code correspond to one protein, right? One amino
Yes, sir.
acids, right? So that means 364 amino acids will be how many nucleotide base? How many bases? That will be 364 into 3. That will result you number of bases, which is approximately, this will be the 364 into 3 means 12 Kshatriya plus Tintia no 1092 means approximately you can go 1090, right? 1090 base pair. This is the basis, right? Now this will be the nucleotide pairs,
Yes, sir.
right?
Yes, sir.
So 1090 nucleotide pairs correspond to this particular protein. Now what you have to do? Now you have to multiply these many bases with average molecular weight of, because you need to calculate what is the duplex DNA required, molecular weight of duplex DNA required. So for that you need to know what is the average weight of one nucleotide base. Okay. What is the average weight of nucleotide base? For one nucleotide base pair, the average weight is 650 Dalton. Okay. And you have total of 190 base pairs. So the molecular weight of this DNA, so the molecular weight of this duplex DNA will be 1090 multiplied by 650 Dalton, right? And that will result you how much? 708500 Daltons. Now, will you be able to calculate the similar question? The answer for will you be able to answer the similar question, right? Is it clear? Is this question clear? Okay. So the next question. Yes. Yes, Arun. Any doubt?
No, sir.
Okay. Okay. So then I will take another question on this topic. Your question is, let me give you another question. Your question is, and these kind of questions are being asked in multiple entrance examination. You will find in CSIR-NET, you will find in your your GATE examination, and also in your end semester examination of this question. If you find a question from this nucleic acid topic, your question will be, your question is question number 2, numerical problem, question number 2. The molecular weight of a bacteriophage T4 double extended DNA. The molecular weight of bacteriophage T4 double extended DNA is 1.3 into 10 raised to 8. Okay, so the question is, question number 1, how many amino acid can be coded for T4 DNA? Question number 1. Second question is, how many different proteins of molecular weight 55,000 Dalton could be coded for T4 DNA? Okay. So how will you approach this problem? How will you approach this problem? This will be the similar to the previous problem that we discussed, right? Arun, can you attempt? To solve this problem, the first thing that you need to know, there is some problem with the internet. Am I audible? Yeah, I hope now the problem is solved. Okay, there is no echo right now. So the question was, the molecular weight of bacteriophage is given T4 double-stranded DNA is given as 1.3 into 10⁸ Dalton. Now, how many amino acid can be coded for T4 DNA? So, how many amino acid? If you want to calculate that, how will you calculate? I have just told you the process of calculating it. First, what you have given? You have been given the molecular weight of T4 double-stranded DNA, which is 1.3 into 10⁸ Dalton. Now, how many amino acids? Before that, you need to calculate how many nitrogenous bases are there, how many base pairs are there. Okay, so for one base pair, your molecular weight will be, so means molecular weight of one base pair will be, I have just told you in the previous question, molecular weight of one base pair is 650 Dalton, right? So how many base pairs are there? Total base pairs in this T4 DNA will be 1.3 into 10⁸. 1.3 into 10⁸ divided by 650. That will give you the number of total nucleotide in this particular sequence, right? And that will be around 10 raised to 2 into 10⁵, approximately 2 into 10⁵ nucleotide pairs. Okay, 2 into 10⁵ nucleotide pairs, and that will be in the coding strand. Okay, now this will be in the coding strand. Now, how many that will be in the coding strand? So now how many codons will be there? So for getting the number of codons, you need to divide this, because 3 nucleotide makes up to one codon. So that means if you divide this particular 2 into 10⁵ by 3, means one codon is 3 nucleotide base pairs. So that means 2 into 10⁵ divided by 3 will give you the number of codons. So that will be 6.7 into 10⁵, 6.7 into 10⁴ codons. Okay, so these many number of codons are there in the T4 DNA. This was the question. How many amino acids? Amino acids means codons, right? So 6.7 into 10⁴ codons are there in the 1.3 into 10⁸ T4 DNA double stranded. So this was the answer for your question number 1. Now for question number 2, you have been asked how many different proteins of molecular weight 550 Dalton could be coded for the T4 DNA. Okay, so for that you need to know what is the average molecular weight of an amino acid, right? So average weight of an amino acid, what is the average weight of amino acid? I have just told you in the previous question. What is the average molecular weight? Arun? What is the average molecular weight of amino acid? Do remember this, because it will help you to solve the problem more efficiently and more accurately in less time. So the average weight of molecular average molecular weight of amino acid is 1110 Dalton, and average molecular weight of a one base pair is 650 Dalton. Remember this conversion. So to calculate number of there are how many total codon, like in this codons, how many what will be the molecular weight? So to calculate that, you need to multiply this with 1110. Okay, so there are now you were asked how many different different proteins of molecular weight of this could be coded for the T4 DNA. So molecular weight is given, which is 550, 55,000. Okay, so the number of amino acid required to make this molecular weight will be 55,000 divided by 110, which means 110, which is 500 amino acid, right? 500 amino acids. So there are total how many codons? 6.7 into 10 raised to 4 codons, and that will give you. So to calculate how many amino acids will be there, how many proteins can be formed based on these codons, and for that you need to divide this 6.7 codons, 6.7 into 10 raised to 4 codons divided by 500 amino acids. For one protein of 55,000 molecular weight, you need 500 amino acids. So if you divide 6.7 into 10 raised to power 4 codons by 500 amino acids, then you will get number of proteins that can be formed of this particular molecular weight, and that will be total of 134. So total of 134 protein of molecular weight 55,000 Dalton can be made from T4 DNA of molecular weight 1.3 into 10 raised to 8 Dalton. Is it clear? Is it clear, or there is any doubt? Arun, is it clear? Have you understood? Have you understood this problem?
Yes, sir.
Okay. Is there any doubt in the middle of the step, or is it completely clear? So if that is clear, now I want to give you few of the facts that you need to remember. So while you are solving, yes. Yes.
Sir, the topic of normality, molarity are included in this subject, sir.
Yeah, that is in the upcoming lecture. While you will be discussing the bioenergetics and all, then normality, molarity, that will come. Okay, even if does not come, then in the next lecture I will take this these all things. If you find difficulty, gives free energy also. Okay, so you need clarity on normality, basically solution part. Normality, molarity, then gives free energy, right?
Yes, sir.
Anything else? Normality, molarity, normality, then molarity, then molality, ppm. These all are the concentration factor. This I will take in the next lecture. Okay, so I will give you few of the important conversion factor in nucleic acid, which will help you in solving the similar kind of problem. The first thing that you need to remember, average molecular weight of DNA base pair is 650 Dalton. Okay. Next thing, 1 AU unit. AU means absorbance at 260 nanometer. 1 AU unit will be equal to approximately 50 microgram per liter of double stranded DNA. Okay. This you will need to solve when the question is given, like this particular A60 units, like A260 units have been given the molecular weight, and then if you want to convert that, you need to the microgram of like molecular weight of the DNA. From there, if you want to calculate the concentration of DNA, this is the conversion. One absorbance of 260 unit is equal to approximately 50 microgram per liter of double stranded DNA. Okay. Then if you want to calculate about the RNA, then absorbance at 260 nanometer unit will be approximately 40 microgram per liter of single strand RNA, ssRNA. Okay. Similarly, one absorbance at 260 unit will be approximately 33 microgram per liter of single stranded DNA. Okay. Now, if you have been given 1000 base pair, 1000 base pairs of DNA in ORF. ORF means open reading frame, means completely it will be coded value. Then if it is in the coding strand and 1000 base pairs of DNA is given, then this will result you total of 330 amino acids. How did you get it? 1000 divided by 3. Okay, approximately 330 amino acid will result you 1000 base pair of DNA ORF. Okay, that means approximately 37,000 Dalton of protein, means 37 kilodalton of protein. So if you have given a protein of having the 37 kilodalton, then it will correspond to a 330 amino acids, and which means it is 1000 base pair of DNA open reading frame. Okay. Now to calculate the concentration of plasmid in a DNA solution, let us say you have been asked to calculate the concentration of plasmid DNA in solution using absorbance at 260 nanometer. Then how will you calculate? You will calculate, the formula will be observed observed A260 nanometer. Okay. Observed value of A260 nanometer. Then multiply it with dilution factor. How many times you have diluted? Multiply it with dilution factor. And then you multiply it with 50 microgram, which means 0.005. Okay, 0.50 milligram. This will give you the concentration in DNA concentration in microgram per ml. Okay. This will be the formula for calculating DNA strength in observed A260. This will give you the DNA concentration in microgram per ml. Is it clear? So these, remember these all conversion formula: first, second, third, fourth, 5, and this one 6th. This is the formula. This is the equation to calculate what is the concentration of DNA in a solution. Plasmid DNA in a solution. Is it clear? Do you want more problem to solve? Let us give you a different kind of problem. Let us solve a different problem. The another problem is, the question is, a sample of purified DNA, a sample of purified DNA obtained from tobacco leaf contains 20 mole percent of cytosine, 20 mole percent of cytosine, assuming that only the 4 principal bases are present, then calculate approximate mole percent of purine residues in DNA. So how will you approach? How will you approach this problem? This is your question number 3. This is a different type from the other 2. How will you approach? you have been given, like in purified DNA, a total from the tobacco leaf. What is the cytosine mole percent? Mole percent of cytosine is 20%. Okay. Now, and you have been given that there are only 4 principal bases present. That means in DNA, which all 4 will be there? A, T, G, C, right? Now, when the cytosine is given as the 20%, that means cytosine will pair with whom? Guanine, right? So the number of cytosine is 20, then the guanine should also be 20, right? So G is equal to C, that means guanine will also be 20. Okay, is it clear? Now, based on this, mole percent of guanine will be also 20%. That means in 100% solution, yes. So in 100%, 13 point. No, you have been asked the purine residue. So purine residue means it is asking to both nitrogenous bases. So normally, if it is following the rule, then A plus G plus T plus C will result you a total of 100%. Is it clear? Is it true? In normal case, adding up to the all four will give you 100%, right? Now I have categorized it as a purine and pyrimidine. Okay, so pyrimidine is T and C, purines is A plus G. Okay, if you add all 4, will result you 100%. Now cytosine and guanine is given. Cytosine, now T plus C, thymine you don't know, T plus 20%. Then A plus guanine is also 20%, will give you 100%. That means A plus T is 100% minus 40, which means 60%. Yes, 60%. Now the number of if G and C are equal, then A and T should also be equal for complementarity, right? For complete complementarity, number of A should be also equal to number of T. That means A plus A is equal to 60. That is 2A is equal to 60. And what will be the A percentage of A? Is 60 by 2. That means 30%. Now, you have been asked to calculate the approximate mole percent of purine residue. Purines are nothing but adenine plus guanine. Okay, adenine plus guanine, right? Adenine and guanine are purine. Thymine, cytosine, and uracil are pyrimidine. So purines means adenine plus guanine. Adenine percentage is 30%. Guanine percentage is 20%. That means both will add up to the 50%. Okay, so purines percentage R here is 50%. Is it clear? Is it clear? Okay, so if it is clear, then I hope this purpose is served. Do you have any further doubt related to this content or the previous content? Okay, So do give the feedback on the portal, NPTEL portal. There is a regular weekly feedback. You can mark your feedback in the portal, and also to the like you can ask your question in your discussion forum. Okay, discussion forum is this one. So you can ask any question in this discussion forum. So whatever the question is there, I will the recordings also I will paste into this portal. Okay, this group, and then you can watch the recording later on if you have missed this content, and all the recordings will be available on my YouTube channel. My YouTube channel is WinBioCB 2021 2022, and this is my YouTube channel. If you go to my channel, do subscribe this channel so that you don't miss out any playlist video. So for biochemistry course, I have uploaded all seven lectures till now. Okay, week 6, week 7, and this one also I will upload. Okay, if there are any further doubt, you can email me, and I will take that question in the next upcoming session. So whatever you have mentioned, that this normality, molality, ppm and Gibbs free energy, these topics I will take up in the next session. So you should join. You must join in the next week session on Friday, same time. I will take these doubts. So if that is it, then I will end this meeting. Thank you for joining. I will upload the recording and then we'll share with all of you. Are there any doubt?